and scientists have no idea where it came from. What they do believe is that if you have that, you may have been evolved from a different kind of species on this planet.
A faint pulse of excitement broke across the scientific community when a research team announced that artificial intelligence had finally cracked one of biology's longest-standing puzzles, the evolutionary and molecular mystery behind blood type O. For decades, the common assumption was that type O, especially O negative, was simply the default or absence version of the ABO blood group system. Researchers described it as a kind of biochemical blank slate, a stripped-down form of A and B. It seemed intuitive enough, but within genetic circles, this explanation never fully satisfied anyone. Too many oddities remained. Too many inconsistencies were brushed aside with just good enough explanations. And the deeper scientists looked, the stranger the whole story of blood type O became. Before we start, smash the like and subscribe buttons for more updates. The breakthrough came from an AI model trained on evolutionary genetics, glycosylation pathways, and millions of biological data points that human researchers had struggled to reconcile. What made this particular model different was not merely its scale, but its capacity to simulate tens of thousands of hypothetical evolutionary timelines side by side, cross-referencing biochemical feasibility with statistical patterns of human migration, pathogen exposure, and ancient population bottlenecks. Where human reasoning reached its limit, the model found patterns no one had noticed and relationships no one had expected. When the system finally generated a coherent explanation, it overturned a century of assumptions in a single, sweeping insight. Blood type O, it revealed, is not the absence of something. It is the presence of a deeply optimized design, an evolutionary refinement so old that it predates modern humans. Rather than sitting at the base of the ABO family tree, as had been believed, type O is the product of intense selective pressures that sculpted it into the most resilient and flexible blood profile nature has produced. This was not a missing antigen, it was a strategically simplified one. Evolution had not erased features from O, it had perfected it. The AI model pulled together molecular details that researchers had previously considered trivial. It demonstrated that the structure of the H antigen, the core molecule underlying type O blood, has subtle conformational advantages that allow for faster cellular recognition, quicker immune response calibration, and dramatically lower risk of inflammatory overreaction. These advantages, while not immediately obvious under a microscope, become unmistakable when simulated across millions of immunological scenarios. The model tested this structure against hundreds of pathogen profiles, ancient and modern, and repeatedly found that populations with higher frequencies of the O genotype survived epidemics that devastated A and B dominant groups. Suddenly, fragments of anthropological puzzles fell into place. The unusual prevalence of type O among certain indigenous peoples in the Americas, the high proportions in isolated Pacific populations, the resilience seen in communities that historically faced intense pathogen exposure, all of it pointed toward O being not a primitive genetic leftover, but an optimized immunological toolset. AI showed that this blood type was refined through cycles of survival during ancient viral sweeps long before written history. What once appeared to be absence was in fact streamlined efficiency. The revelation forced a re-examination of assumptions embedded in medical training for generations. The idea that O negative is simply universal donor due to lacking antigens now seemed incomplete. Instead, AI demonstrated that the universal compatibility arises from structural neutrality designed not out of deficiency, but out of evolutionary strategy. The antigen profile of type O appears to act like a universal conversational language for the immune system, uncomplicated, adaptable, and far less likely to trigger defensive overreaction in foreign environments. This made type O individuals, particularly O negative, uniquely valuable in emergencies, but it also hinted at deeper biological functions that had been missed. One of the most surprising insights came from simulations of early human migration. According to the model, the extreme mobility of early human groups, combined with exposure to vastly different ecological environments, favored a blood type that minimized immunological friction. The more populations moved, interbred, and exchanged pathogens, the more an adaptable blood profile would have been favored. In this context, type O's rise was not random. It was a decisive evolutionary advantage. AI traced this back through reconstructions of prehistoric pandemics, identifying an era in which O carriers had a statistically significant survival edge that shifted the entire genetic landscape of early humanity. The discovery challenged not only scientific assumptions, but cultural ones. For centuries, various societies attached folklore and stereotypes to blood types, especially in parts of East Asia, where personality traits were once superstitiously tied to A, B, AB, and O. While these beliefs held no scientific weight, the new findings brought a sense of poetic irony. Type O, long seen as the simple or basic type, was in fact the most refined outcome of human ancestry's battle with disease. It was a testament to survival against overwhelming odds encoded into the bloodstream itself. The AI model did more than trace evolutionary patterns. It reconstructed the molecular journey of the gene responsible for the ABO system, mapping out how subtle mutations shaped the antigen pathways. It showed that the O genotype, far from being a loss of function, evolved through a series of mutations that streamlined the glycosyltransferase enzyme, reducing unnecessary complexity. This simplification wasn't accidental, it improved metabolic efficiency and reduced the risk of misfolded proteins, an advantage that grows over thousands of generations. For a mobile species facing extreme environmental variability, such efficiency could mean the difference between life and death. These findings shifted the focus of many immunology research programs. Scientists began exploring how the structural advantages of type O might inform vaccine development, inflammation management, and autoimmune disorder prevention. The adaptability and minimal antigenic interference of the O profile offered clues for designing more universal therapies. If nature had already created a model for broad immunological compatibility, researchers wondered what lessons it could teach about reducing adverse reactions in medical treatments. The implications stretched even into transfusion science. Doctors had long used O-negative blood in emergencies due to its compatibility, but now attention turned to whether replicating the O-like simplification artificially might produce safer synthetic blood alternatives. AI models began simulating synthetic antigen profiles based on the principles seen in type O, raising the possibility of engineered universal blood that could be manufactured in labs, consistent and safe across all patients. Excitement also grew in evolutionary biology. With this newfound understanding, researchers revisited questions about ancient human interactions with extinct hominins. DNA extracted from Neanderthals and Denisovans showed variations within the ABO system, but previous interpretations were limited by the assumption that type O was primitive. Now, AI allowed scientists to reinterpret how these ancient relatives interacted with early humans, and whether their pathogen exposures contributed genetic advantages that helped shape the rise of type O. New hypotheses emerged about how interspecies gene flow influenced immunological evolution, expanding the story of humanity's ancestry. Some anthropologists turned their attention to patterns of settlement and survival in prehistoric societies. If type O carriers were more resilient during specific epidemics, this may have influenced the geographic expansion of certain groups. Regions with higher O prevalence today might reflect ancient survival corridors, routes where populations endured outbreaks that eliminated others. AI models began reconstructing these routes, layering genetic data over climate models and archaeological records. It painted a dynamic portrait of human survival, tribes navigating not only landscapes of land and sea, but landscapes of disease. The discovery also touched on the nature of biological classification. Scientists had long treated blood type O as a lack of certain antigenic markers, a kind of immunological blank checkbox. But the AI model reframed blood groups as strategies, each with strengths and vulnerabilities. Type A may respond more efficiently to certain pathogens. Type B may offer advantages against others, but type O emerged as the strategy designed for unpredictability, robust, flexible, and broadly defensive. This suggested that the variety within human blood groups was not a flaw or a quirk of genetic drift, but a mosaic tapestry of survival tactics honed by different environments and challenges. Even within medicine, the revelation encouraged a more nuanced understanding of patient variation. Doctors had long known that blood type can affect susceptibility to certain diseases, such as clotting disorders or particular infections. But the discovery prompted them to consider how deep evolutionary pressures might influence these patterns. It raised questions about whether type O's evolutionary optimization could be linked to certain modern vulnerabilities, such as slightly increased bleeding risk, and how these traits may have been trade-offs shaped by prehistoric environments. AI also highlighted that the story of blood type O was still unfolding. Evolution never stops, and the pressures shaping humanity today are different from those faced by ancient populations. It raised the possibility that new selective pressures, modern epidemics, global travel, environmental changes could continue to reshape the ABO landscape. If type O was optimized for a world of constant movement and pathogen diversity, how might it fare in a world with advanced medicine but new global threats? Researchers found themselves facing fresh questions about how the ABO system might evolve in the next 1,000 years. The announcement triggered renewed interest in the ethics of genetic research. Understanding the evolutionary strengths of type O raised concerns about how such knowledge could be misused. Discussions emerged about ensuring that discoveries about blood groups remain grounded in medical value, not misinterpreted as indicators of superiority. The scientific community emphasized that every blood type brings unique strengths shaped by distinct paths of human history. The revelation about type O was a reminder of complexity, not hierarchy. Yet despite these concerns, the sense of wonder remained. The AI-derived explanation carried an almost story-like quality, turning what was once thought of as biological absence into a symbol of ancestral resilience. People who had never thought much about their blood type found themselves suddenly fascinated by what it implied about their lineage. O carriers in particular began to see themselves not as bearers of a blank, antigenic state, but as inheritors of a deeply ancient adaptation, a kind of molecular passport stamped with episodes of survival older than civilization. The research also reshaped how scientists approached AI in biology. This was the first time a model had successfully integrated such diverse datasets into a unified evolutionary narrative. The achievement demonstrated the growing potential of AI not just to analyze data, but to generate novel hypotheses that human minds alone might never conceive. It encouraged researchers to apply similar systems to other biological enigmas. The origins of certain immune responses, the genetic basis of longevity, the unexplained quirks of human metabolism. If AI could decode the hidden logic behind something as complex and ancient as the ABO blood system, what else might it illuminate? As the months passed, teams around the world began testing the AI model's predictions in laboratory settings. Early results confirmed many of the structural advantages it described in the H antigen. Experiments showed measurable differences in antigen folding stability, enzymatic efficiency, and immune modulation. Some labs began exploring whether these advantages could be harnessed in biotechnology, such as designing cell lines that mimic O-like antigen simplicity to reduce rejection in therapeutic treatments. The medical implications continued to expand. The compatibility principles underlying type O offered clues for reducing organ transplant rejection, a persistent challenge in healthcare. Researchers speculated that if the immune system responds more calmly to O-like profiles, then engineering tissues with simplified antigen patterns might improve transplant success rates. This idea, once purely theoretical, now had a mechanistic foundation thanks to the AI model's detailed breakdown of antigen-immune interactions. Another avenue of research focused on infectious disease modeling. If type O evolved as a response to pathogen pressure, understanding its advantages could help identify vulnerabilities in future pandemics. Scientists began using the AI system to simulate how different blood type distributions might influence the spread or severity of emerging viruses. These simulations allowed public health experts to refine strategies for outbreak response, taking into account genetic factors that had previously been overlooked. Throughout academia, the findings sparked a reevaluation of long-standing assumptions. Textbooks were rewritten, conferences dedicated sessions to the unraveling of the O mystery. Students entering biology programs found themselves learning a narrative that previous generations had never encountered, one that integrated genetics, anthropology, biochemistry, and AI into a single cohesive story. It was a new way of understanding how human biology is shaped not only by mutation and natural selection, but by vast networks of interacting forces that AI was uniquely capable of analyzing. Interest in the discovery spilled into public life as well. Discussions about ancestry and human migration gained a new dimension, with people curious about how their blood type connected them to ancient events. Museums curated exhibits explaining the evolution of blood groups, presenting the revelation not as a distant academic finding, but as a living part of every visitor's biology. Educational programs highlighted how AI can uncover secrets hidden within the human body, inspiring the next generation of researchers. The deeper the scientific community delved, the more the discovery revealed about humanity's collective past. The story of Type O became a story of endurance, adaptation, and the relentless pressure of survival. It painted a picture of ancient populations navigating landscapes filled with unseen threats, where the tiniest molecular configurations determined the difference between flourishing and extinction. It reminded people that evolution does not sculpt with intention but with consequence, leaving behind structures that carry the imprints of countless triumphs and struggles. What made the AI-decoded explanation so compelling was not only its scientific precision, but its narrative coherence. The shifting landscapes of disease, each piece fit into a larger mosaic that was only visible when seen through the vast interpretive lens of artificial intelligence. And beneath all of this, the discovery invited a quiet moment of reflection. Inside every O carrier, and indeed inside every human carrying any blood type, is a story that began long before birth, long before cities or civilizations, long before language itself. A story of ancestors who endured unimaginable challenges and passed forward tiny molecular arrangements that helped the next generation survive. For Type O, the AI model revealed how those arrangements were not leftovers or omissions, but evolutionary signatures of remarkable resilience. The mystery had never been about what Type O lacked, but about what it represented. And thanks to AI, that representation became clear. A design shaped by ancient pressures, refined by survival, and encoded into the blood of millions across the world. What seemed simple was in fact profoundly intricate. What seemed empty was full of history. And what seemed like an absence was, astonishingly, a masterpiece of evolutionary engineering. What the AI uncovered about the origins and purpose of Type O blood opened doors to questions that scientists had never thought to ask. The discovery had solved one mystery, but in doing so had revealed an entire ecosystem of hidden biological strategies woven through human history. Researchers quickly realized they were examining only the first layer of a far deeper story. One of the most intriguing threads emerged from the AI's ability to model molecular behavior inside extreme environments, something that traditional biological experiments struggled to replicate. The system simulated antigen behavior under heat stress, nutrient scarcity, high-altitude oxygen variation, and prolonged immune activation. In these simulations, Type O consistently demonstrated greater molecular stability than types A, B, or AB. This wasn't just about resisting pathogens anymore, it was about endurance at the cellular level. This revelation prompted scientists to revisit long-standing anthropological puzzles involving ancient settlements in harsh climates. Populations living in high-altitude regions, arid deserts, or dense tropical rainforests often exhibited higher proportions of Type O blood. Previously, this pattern had been explained away as coincidence or founder effect, the random result of small populations carrying certain genetic traits. But the AI's findings hinted at a more intentional evolutionary story. The simplicity and adaptability of Type O antigens may have offered subtle advantages in environments where the body was under constant physiological stress. In tropical regions, where parasites and viruses flourish, The immune system must remain alert, but not overreactive. Overreaction can be just as deadly as infection. The AI model showed that the streamlined antigenic structure of type O reduces the likelihood of immune misfires, allowing for quicker recalibration during a pathogen assault. In contrast, the more complex antigen structures of A and B may have occasionally triggered unnecessary inflammation under extreme pathogen loads, which could become fatal in environments with limited access to healing resources. At high altitudes where oxygen levels fluctuate dramatically and oxidative stress is constant, the model revealed that type O's simplified antigen chemistry reduced the metabolic cost of maintaining red blood cell integrity. Even tiny differences in metabolic efficiency compound over thousands of generations, potentially offering survival advantages in extreme terrains. This finding gave new context to the prevalence of type O among populations in the Andes and parts of the Himalayas. As researchers pieced these insights together, they began to consider how blood type shaped not just biological survival, but cultural development. If certain blood types conferred subtle environmental advantages, might they have influenced migration choices, settlement patterns, or even the success of specific ancient societies? It was a bold idea, but the AI's data simulations offered tantalizing clues. For example, early agricultural societies in stable environments may have favored individuals with type A or B, whose antigenic complexity could provide heightened immune precision against certain localized pathogens. Meanwhile, nomadic groups facing unpredictable threats might have unintentionally selected for type O individuals, whose broader immunological versatility made them more resilient travelers. This proposed interplay between blood type distribution and cultural evolution reshaped discussions in both anthropology and genetics, opening avenues for interdisciplinary research that had previously been unimaginable. Another unexpected direction of inquiry came from energy economics within the human body. The AI model calculated that the enzymatic efficiency associated with type O reduces the metabolic cost of antigen synthesis over a person's lifetime. These savings are small on a day-to-day scale, but significant when scaled across entire populations for centuries. This raised the hypothesis that in prehistoric times, when food scarcity was common, individuals with type O may have had marginally better energy resilience, contributing to higher survival rates during famines or long migratory journeys. It positioned type O not just as a blood profile, but as a metabolic strategy tuned for human hardship. The implications extended into modern medicine as well. As scientists examined how antigen simplicity interfaces with inflammation pathways, they noticed parallels with autoimmune conditions. Complex antigenic profiles can, in some circumstances, create higher risks of misidentification by the immune system. This led researchers to investigate whether type O's reduced antigenic complexity could offer clues for designing therapies against autoimmune disorders. By studying how type O antigens avoid triggering chronic inflammatory responses, scientists hope to design interventions that mimic these mechanisms in individuals of other blood types. Meanwhile, biotech innovators began exploring how the AI's findings might transform gene editing research. If type O represents a streamlined optimization of the antigen pathway, could targeted gene editing reproduce some of those efficiencies in other blood types without fully converting them to O? The possibility of reducing rejection rates in cell-based therapies or simplifying antigen expression in engineered tissues sparked new collaborations between geneticists and medical engineers. The goal wasn't to erase genetic diversity, but to learn from the elegantly optimized architecture of type O. One of the most forward-looking applications emerged from synthetic biology. The AI's antigen simulations provided a template for creating simplified antigen systems for artificial blood substitutes. Researchers had long struggled to produce synthetic blood that could safely circulate in any human body without triggering an immune reaction. The insights gleaned from type O began shaping new molecular designs for synthetic red blood cells, built on principles of antigenic minimalism. These designs aimed to produce blood substitutes that would bypass immune recognition hurdles entirely, reducing dependence on donor supplies and potentially transforming emergency medicine. The discovery also propelled deeper inquiries into prenatal development. Blood type interactions between mother and fetus have always played a role in pregnancy outcomes, especially with Rh incompatibility. Scientists began to investigate whether the evolutionary emergence of type O influenced maternal-fetal immune dynamics. Early models suggested that the simplified antigenic profile might have contributed to lower rates of immune-triggered pregnancy complications in ancient populations, subtly improving reproductive success during periods of environmental instability. Across research fields, the AI's findings inspired rethinking of long-held assumptions. Even the concept of universal donor took on new meaning. What had been treated as a clinical convenience now appeared as a sophisticated evolutionary design. The more scientists learned, the more they marveled at how much complexity had been hidden behind such a simple label. As inquiries unfolded, interest grew in the genetic pathways leading from ancient blood types to the modern ABO system. The AI traced ancestral glycosyltransferase enzymes far deeper in evolutionary time than expected, finding parallels in distant primate lineages and even earlier mammalian branches. This insight challenged the notion that blood types were relatively recent evolutionary developments. Instead, it suggested that the ABO system may have roots extending tens of millions of years, shaped by cycles of disease far older than humanity itself. This raised profound questions about the role of blood types across mammalian evolution. If type O-like simplification provided survival advantages under certain conditions, might similar evolutionary strategies have appeared independently in other species? Comparative genetic analysis revealed tantalizing patterns. Ancient mammals facing intense environmental fluctuations often exhibited blood group systems that trended towards simplified antigen structures. These patterns suggested convergent evolution, a process by which unrelated species evolve similar traits when facing similar pressures. The AI's work hinted that nature repeatedly gravitates toward antigenic simplicity as a durable strategy in chaotic environments. The discovery that type O may represent a refined, convergent solution rather than a mere absence reshaped evolutionary narratives. It also prompted philosophical discussions about what optimization means in a biological context. Evolution does not aim for perfection, but for survival. The fact that type O emerged through simplification rather than elaboration became a lesson in how efficiency often outperforms complexity when survival is at stake. As scientists prepared to integrate these findings into broader theories of human evolution, another line of inquiry emerged: the relationship between blood type and the microbiome. The AI model suggested that antigen structures influence which bacterial species flourish on mucosal surfaces, including the gut. Researchers had known that blood type could affect intestinal flora, but the AI predicted far deeper, more systemic interactions. It proposed that type O's antigenic neutrality may create a more flexible environment for diverse microbiome compositions, which in turn could have contributed to nutritional adaptability during ancient migrations. This idea held immense significance. A resilient microbiome could help humans digest unfamiliar foods, extract nutrients from scarce resources, and resist novel pathogens. If type O fostered this adaptability, it could have been a silent companion to early human explorers crossing continents and oceans. Understanding this relationship might offer insights into nutrition science and gastrointestinal disease treatment today. Meanwhile, epidemiologists began using the AI's models to examine how blood type distributions might influence global disease patterns. Although no blood type grants absolute protection, type O's adaptive immunological features could contribute to different outcomes during certain outbreaks. This does not imply invulnerability or superiority. Instead, it highlights the subtle ways in which evolutionary history shapes the present. Epidemiological models that integrate blood type distributions could become valuable tools for anticipating how certain pathogens may spread or mutate in the future. The discovery also prompted questions about genetic diversity and balance. If type O offered so many advantages, why did types A, B, and AB persist? The AI provided an intriguing answer: evolutionary equilibrium. each blood type offers different immunological strengths. While type O excels in versatility, types A and B show enhanced responses against particular pathogens. This balance prevents any one blood type from dominating entirely, preserving genetic diversity that strengthens humanity's overall resilience. It is a reminder that every genetic variant carries its own history and its own purpose. As researchers probed further, they explored whether the rise of type O corresponded to specific turning points in human evolution. The AI identified several epochs where pathogen-driven selection intensified, possibly shaping the spread of type O. These moments included ancient viral sweeps, climatic shifts that altered disease ecosystems, and population bottlenecks that forced small groups to adapt quickly. Type O's stability and versatility may have been crucial during these periods, helping communities survive when complexity became a liability. The more scientists learned, the clearer it became that the story of type O was inseparable from the story of humanity. Not because it was superior or more important, but because it was a reflection of how survival shapes biology in ways that are not immediately visible. It symbolized the principle that simplicity, when refined by necessity, can become one of evolution's most powerful tools. As the implications of the AI's discovery continued to unfold, excitement grew around potential applications in regenerative medicine. Tissue engineers began investigating how simplified antigenic signatures could be used to create universal cell lines for therapies. If cells could be engineered to mimic the immunological neutrality of type O, they might avoid rejection in patients of any blood type, vastly expanding access to stem cell therapies and engineered tissues. Another frontier opened in personalized medicine. The AI's frameworks suggested that blood type could be integrated with genetic ancestry, metabolic patterns, and microbiome profiles to create more tailored medical treatments, not by assigning stereotypes to blood types, but by understanding how different antigenic strategies interact with broader physiological systems. Such personalized approaches could improve treatment outcomes in everything from infection management to chronic disease prevention. Philosophers and historians of science joined the discussion as well, intrigued by how deeply assumptions can shape scientific narratives. For more than a century, type O had been taught as the absence of A and B. This wasn't a malicious simplification, it was simply the most intuitive explanation available with the tools of the time. The AI's revelation served as a powerful reminder that scientific understanding evolves, and that even the most familiar biological concepts may hold hidden depths waiting to be uncovered by new technologies. The discovery also highlighted a timeless truth about human curiosity. The ABO system had been mapped genetically, chemically, and medically for decades, yet a fundamental misunderstanding persisted because the deeper patterns lay hidden beneath layers of complexity. AI didn't replace human reasoning. It illuminated connections that humans could then interpret and explore. It became a tool for expanding human understanding, turning mysteries into gateways for new ideas. If you liked this video, please give it a like and subscribe to our channel. Also, leave your comments below and tell us, what are your thoughts on this AI discovery about blood type O predictions below? We want to hear from you. Thank you for watching and see you next time.